So we have talked about different types of culture medias. The anaerobic culture media also have been discussed, and we also discussed about different inoculation methods. Now come to the culture identification. Okay, so we have inoculated the specimen, the and then after inoculation, we have incubated the culture media, and we have seen the colonies growing over that culture medias also. After the colonies growth has occurred on the culture media, we have to identify the microorganisms, and that will be discussed here. How are we going to identify the microorganism in that culture media or in that colony? So that is we will be discussing here in the culture identification topic. So let's discuss about the culture identification. So we have the two methods of culture identification, that is the conventional biochemical test and the automated systems. Now first come to the automated systems. So automated systems are quick, are more accurate, and here in automated system we can do AST also along with identification. Okay, so in alongside, while doing identification of the microorganism, the antimicrobial susceptibility testing is also done. While in case of conventional methods of identification, we have to separately do antimicrobial susceptibility testing, but that's not the case with the automated systems. Okay, so definitely the automated system has reduced our burden and has increased our accuracy in the recent times about the detection of the microorganisms from different specimens or from different cultures. The examples of the automated systems are the Malditov and the Vitek. You must have been seen these Malditov and the Vitek etc. systems in your microbiology labs. I mean, in our college in NRS MCS, there is definitely this Malditov and Vitek machines in our microbiology labs, and is used for the identification of the microorganisms and also for the antimicrobial susceptibility testing. After that automated system, we are not going to discuss about that automated system in detail. I mean, that is not so important for us, but obviously this conventional biochemical tests are very important to discuss. So the different examples of the conventional biochemical tests are the oxidase test, the catalase test, oxidase test, the citrate utilization test, urea hydrolysis test, the indole test, and the triple sugar iron test. We will discuss about these different types of conventional biochemical test one by one. But before discussing about the different principles, the methods, and the interpretation of these tests, let's first talk about the examples of the organisms which are catalase test positive, which are oxidase negative or oxidase positive, which are citrate test positive or citrate test negative, which are urease positive or urease negative, or indole positive or indole negative as a whole. So when we talk as a whole, then we will be able to memorize it for a longer duration, and that I remember by a score which is called as the Kokui score, COCUI score. So we will just remember the Kokui score for different microorganisms, and by that we will be able to remember the whether the organism is a particular test positive or negative. So COCOI stands for catalase, then O for oxidase, so C O for oxidase, then C for citrate, U for urease, and I for indole. So this is our COCOI score. So in the COCOI score, let's talk about the most important microorganisms that we talk in day-to-day life talk about in day-to-day life. So first of all, we have the pseudomonas. Pseudomonas is catalase positive, oxidase positive, citrate positive, urease negative, and indole negative. So we are seeing that the Kokoi score of this pseudomonas is P3N2. So P3 means first three tests are positive, and N2 means last two tests are negative. So see here, positive, 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 and last two are negative, negative, negative. So similarly, E. coli is positive, negative, 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 and positive. That means the Kokoi score of this E. coli is PN3P. PN3P. So first catalase test is positive, then N3 means oxidase, citrate, and urease tests are negative. Oxidase, citrate, and urease tests are negative. That is N3. And again, at last we have P. That means indole test is positive. And for Klebsiella, we are seeing that the the catalase test is positive, then oxidase is negative, then citrate is again positive, urease is positive, and indole is negative. That means here the cocoa score is pnp2n, pnp2n. Also for proteus we are seeing that the cocoa score is pnp2n, and for vibrio the cocoa score is p2vnp. Here is a small the difference, that the citrate test for Vibrio cholerae is variable. That means it can be positive also, it can be negative also. Uh, uh, so that causes a slight change in our cocoa score, that is P2V and P2V and P. And for Salmonella, the cocoa score is P and 4. That means the site catalase is positive, and then last four tests, the oxidase, citrate, urease, and indole are negative. That means P and 4. So if you are going to remember the different tests for different bacteria separately, I, I mean one by one, then I'm sure that you are going to messing up in your exams. So this is my way to remember the tests results of different bacteria. So for pseudomonas, the cocoa score is P3N2. Just remember P3N2 and you are done with all the tests. You know all the results of the test. P3N2 means the catalase, oxidase, citrate will be positive, urease, indole will be negative. Simple. For protease, suppose for protease, if someone asks you like what is the citrate test for protease, then if you remember that the cocoa score of the protease is p and p2n, then you can say that C comes third, and third here later is p2 means citrate and urease will be positive. That means citrate will be positive for the protease. You will immediately say that citrate test for protease is positive. So you have nothing to do uh, extra. You just remember the cocoa score. Okay, just cocoa score should be remembered, and by that you remember all the taste of these six bacteria, the most commonly seen bacteria or most commonly studied bacteria. Okay, and by this you remember all the results, I mean all the examples also, na. So this is serving two purposes. Okay, you need not to remember the examples of catalase positive, oxidase positive, citrate positive, or urease or indole positive. You just remember the cocoa scores. You will be remembering, I mean you can be able to tell the catalase, oxidase, and citrate, urease, indole positive or negative examples. Okay, so you need not to bother about that example, just remember the cocoa score. Okay, now coming to the different tests separately. So first of all, we'll talk about the catalase test. So in the catalase test, what we do is that we mix the bacterial colony with 3% hydrogen peroxide on a slide, and then we wait. As we wait, we see that bubble is produced. If bubble is produced, that means it is catalase positive. That means how is that bubble is producing? So let's see the principle also. The principle is that the catalase producing bacteria produce the catalase enzyme. This catalase enzyme causes the breakdown of the hydrogen peroxide into water and oxygen. And as this oxygen is produced, this causes these bubbles to get produced in that, over that slide. So those bubbles are nothing but the oxygen bubbles. Okay, and as these oxygen bubbles are getting produced, that indirectly indicates that catalase is produced by that particular bacterial strain. Okay, and thereby we can say, we can identify the catalase positive organisms. So bubbles are indicator of the catalase positive organism. Next is the oxidase test. So in the oxidase test, first see the principle. The principle of oxidase test is that the, here we identify the cytochrome oxidase producing bacteria. In the oxidase test, we identify, or we detect the cytochrome oxidase producing bacteria. How is that possible? So this cytochrome oxidase causes the oxidation of the oxidase reagent. Now, if you want to remember the name of that oxidase reagent, that is the tetramethyl paraphenylenediamine dihydrochloride. That's such a big name. So, if you want to remember the name of the oxidase reagent, you can remember, but I do not remember the name. The simple way to remember is that oxidase reagent. So when this enzyme acts on that oxidase reagent, which is nothing but tetramethyl paraphenyl and diamine dihydrochloride, then a purple colored compound is produced, that is indophenol blue. Now do not get confused from the name indophenol blue. Although the name contains blue, but it is purple. So oxidase reagent, see here, when acted upon by cytochrome oxidase produces the indophenol blue and the purple color is produced. That means if the purple color is getting produced, we can say that the bacteria is producing oxidase enzyme. That's when the oxidase reagent is getting converted to indophenol blue and causing the production of that purple color. So whenever the purple color is produced, that means the bacteria is oxidase positive. And if there is no color produced, this is not always yellow, so please correct it. This is not always yellow. So even if even if uh, no color change is seen, or even if yellow color is produced, then it will be oxidase negative. Now coming to how are we going to do this, we know the principle behind that test, but how are we going to do that? So what do we do is that we take a paper strip. Okay, we will soak it into that oxidase reagent. Now tell me the name of the oxidase reagent. That is the same tetramethyl paraphenyl and diamine dihydrochloride. Okay, so that paper strip is soaked in that oxidase reagent, and then we will inoculate the bacterial colony over that paper strip, and then we will wait for 10 seconds. If we are seeing this purple color, that means it is a oxidase positive organism. It is a oxidase positive organism. If we are seeing no color change, that means it is a oxidase negative organism. Now coming to the citrate test. So in the citrate test, now let's first also see the principle of the citrate test. So the principle of citrate test is that the citrate utilizing bacteria utilize the citrate for as a carbon source. Whenever organism is producing some food, it must need carbon. We also need carbon. Plant also need carbon to make the carbohydrates. So the citrate utilizing organism use the citrate as a carbon source for preparation of their food. Now, when they use the citrate, they also produce some alkaline byproducts unknowingly. Okay, so when they produce these alkaline byproducts unknowingly, we use these features to identify those organisms. So this feature, how are we going to use this alkaline byproducts? So these alkaline byproducts, when produced, they change the color to the, I mean the alkaline pH is more than 7 na. So that alkaline pH will cause the change of the color of the media. So we do this citrate utilization test in the Simon's citrate medium, which is green in color. So as the pH increases due to production of that alkaline byproduct, the color, the green color changes to blue, and whenever the blue color is produced, that means it is a citrate positive organism. So see here, we have citrate media which is green, alkaline byproduct is produced, and the color is changed to blue color due to pH more than 7 due to alkaline medium. So interpretation is that if the blue color is produced, it is citrate positive. If there is no color change or it remains green itself, then it is negative, because there is no color change in the in the media, because alkaline byproducts would not have been produced by that organism. That means the bacteria is not a citrate utilizer bacteria. That means it is a citrate utilization negative bacteria. Now coming to the method, how are we going to do that citrate test? So we inoculate the bacterial colony on the Simon's citrate agar, and then we incubate it and wait till the color is changed. Usually after overnight incubation, the color change is seen. If the color change is not seen, that means it is a citrate negative bacteria. Simple. Now coming to the urease test. So urease test has a urease test, in the principle of the urease test is that there is urease producing bacteria. If the bacteria produces the urease enzyme, that urease will break down the urea to ammonia and carbon dioxide. Now that ammonia is alkaline, we all know that ammonia is alkaline. So that alkaline means pH is more than 7 again. And in that urea agar medium where we do this urease test, that also contains phenol red as an indicator. That also contains the phenol red as an indicator. So when the pH is increasing more than 7, it is becoming alkaline. Then that phenol red indicator changes to pink color, and if we are seeing this pink color, that means the organism is urease positive. See here, pink color means urease positive. And if there is no color change or yellow color is seen, that then it is urease negative bacteria. How do we do this urease test? So we inoculate the bacterial colony on the Christensen's urea media. I have talked about this Christensen's urea media in our culture media lecture number 3, that means part 3, where I discussed this Christensen's urea media as a differential media. And there also I have told that it contains phenol red as an indicator, and the urease producing bacteria causes the change in color of the media to pink color. And this feature is utilized in the urease test, and we identify the urease producing bacteria. Okay, so after inoculating the bacteria colony on the Christensen's urea media, we incubate and wait till this pink color is produced. If the pink color is produced, that means it is urease positive bacteria. If no color changes in, then it is a urease negative bacteria. So till now we talked about the catalase test, the oxidase test, the citrate test, the urease test. Now we'll talk about the indole test. So the indole test, the principle of the indole test is that in the indole test we identify or we detect the tryptophanase producing bacteria, the tryptophanase enzyme producing bacteria. What does that tryptophanase do? That tryptophanase, by the name itself we can get to know that the tryptophanase will break down the tryptophan, which is an amino acid. So the tryptophan is broken down into indole by tryptophanase. See here, tryptophan is acted upon by tryptophanase and indole is getting produced. Now that indole forms the forms some complexes with the coax reagent, that forms some complexes with the coax reagent when the coax reagent is added. So till here the coax reagent was not added. So once we incubate that incubate that media overnight, then uh, after one night we add the coax reagent over that. And if the cherry red color ring is produced at the surface of that bacterial broth, that means it is indole positive, because this coax reagent can make the complexes only with indole, and indole will be produced only if the bacteria is a tryptophanase producing bacteria which has broken down the tryptophan to indole. So that means indirectly we can say, if the cherry red color ring is produced, indirectly we can say that the bacteria is a tryptophanase producing bacteria. And we say that the bacteria is indole positive. If yellow coloring is in, that means we say it as the indole negative bacteria. That means no ring is produced, that means indole was not produced. And if indole was not produced, then it is a indole test negative bacteria. Okay, and why indole is not produced? Because the bacteria was not able to produce the tryptophanase enzyme. Okay, so that's the meaning of the indole negative. How are we going to do this indole test? So bacterial colony here is inoculated into tryptophan containing broth, and that broth is incubated overnight. After incubation overnight, we add the coax reagent. So till now, here in this part we did not have the coax reagent, please mind this. Okay, we did not have the coax reagent in this part till incubation overnight. Okay, only after incubation overnight we add this coax reagent. And this coax reagent is a very big name reagent. If you want, you can google it. Okay, and if you want to remember, then remember it from google. Okay, I'm not going to tell about this coax reagent. That's a very scary name about that coax reagent. So after adding that coax reagent, we will wait till this cherry red color ring is produced. If it is produced, then it is a indole positive. If yellow red color ring is produced, then it is a indole negative. Now we'll see some of the pictures. So let's see here. This is our catalase test. See here, oxygen bubbles. We are able to see the oxygen bubbles produced over that slide. That means this is catalase positive, and here no oxygen bubble is seen. That means it is catalase negative. So catalase positive means oxygen bubbles are there. Negative means no oxygen bubble is there. See here, oxidase test. Let's talk about the oxidase test. So in the oxidase test, we have this strip. We have this two strips. In the strip one, this is strip two. So in the strip one, we can see the purple color change is produced. Purple color change is produced. That means it is a oxidase test positive bacteria. And no color change is seen in strip two. That means it is oxidase negative. Next, come to the citrate test. So see here our citrate test. This is our citrate test. So in our citrate test, we were using the Simmons media. So Simmons media color is green. This is see the Simmons media which is green in color. Okay, this is Simmons media green in color. And if the organism is a citrate utilizing bacteria, or organism is a citrate utilizing organism, then it will be producing the alkaline byproduct which will be converting this green color to blue color, which will be converting this green color to blue color, this green color to blue color. So that means if we are seeing this blue color in the Simmons green medium, then we can say that it is citrate positive. And if no color change is seen, that means it is a citrate negative. See here the urease test. So in the urease test, we saw that if the pink color is produced, then it is urease positive, because urease breaks down the urea to ammonia and CO2. Ammonia is alkaline. That alkaline causes pH of more than 7, and more than 7 pH causes the phenol red to change the color to pink color. So as the pink color is produced, that means it is urease positive. Here no color change is seen, that means it is urease negative. Or yellow color is seen, that means it is urease negative. Now coming to the indole test. So this is our indole test. Okay, this is our indole test. See here, this is bacterial broth. This is bacterial broth. This is also bacterial broth. We have added the coax reagent after the bacterial over that bacterial broth after overnight incubation, and we can see here the cherry red color ring at the surface. So this ring is suggesting that it is a indole positive. This is indole positive. This is indole positive. While here we can see the yellow ring, that means it is indole negative. It is indole negative. So this is our all about this conventional biochemical test, and this is the all the pictures of all the tests. So I have compiled this whole tests, the biochemical conventional test in 2 pages to make it simpler to remember. And the principle is generally asked in your microbiology vivas, and also the principle is asked as a MCQ also in the national board exams, in the in the NEET PG and all those exams. So principle should must be remembered. And please remember this COCOI scores, because this COCOI score will help you remember the which bacteria is catalase positive, or which is negative, or which is citrate positive, citrate negative, or indole positive, or indole. So you can give the examples also by remembering this COCOI score. Please remember this COCOI score. This is very important. Okay, and very easy also, and you will not make any mistake if you remember this COCOI scores. Okay, while telling the results of different bacteria. So this is all about the culture identification.